You need constrained chemistry. As John Sutherland says, you can't use what comes in on meteorites. So when people write to me, oh, well, meteorites provide this, meteorites provide. Well, first of all, they're not providing the stereo-controlled compounds that you need, but also there's millions of different compounds on those meteorites so that it becomes useless. People don't know it's there. If it's there, you can use it. No, you can't.
I do understand what you're talking about. There is a very strong community that is protecting the status quo. And the thing I found out early on is if you go to any college or university, the biology faculty always outnumbers the chemistry faculty. So there's more of them than there are of us. And it's just the organic chemists that have the expertise to see the folly in some of these abiogenesis schemes.
Welcome. We have with us today a guest that I'm really excited about speaking with. He and I have never met before, and so this is the first time we're meeting. We've corresponded a few times by email recently, and we were introduced together through our friend Bill Dembski. And Bill wrote on his Substack an interview with Ed, and now we've got Ed with us. Let me just read the background of Ed Peltzer, and he's a seasoned ocean chemist, and he has a long and profoundly interesting. He's been profoundly interested in the origin of life. He did his doctoral studies at the Scripps Institution of Oceanography, SIO, and he was mentored by two luminaries who I'm sure you've heard of. Well, the first one I'm sure you've heard of, his name was Stanley Miller of the famous Miller-Urey experiment. And so that was his PhD mentor. And that was an experiment simulating early Earth conditions for the formation of amino acids. And his other mentor was Jeffrey Bada, an expert in the field of amino acid racemization and a prominent figure in the study of organic compounds and meteorites, which Ed Peltzer has worked on himself as far as his PhD. So this has deeply influenced Ed's perspective on abiogenesis. He's been engaged in discussions and presentations on the complexity of life's emergence, often emphasizing the challenges inherent in explaining the transition from inorganic, non-living matter to organic matter that could then generate life. And his collaborations and dialogues with leading scientists in the field underscore his commitment to exploring the fundamental scientific questions. And he is an ocean chemist, but he's a specialist, and he was the senior research specialist and chemical safety officer at the Monterey Bay Aquarium Research Institute, which I have actually been to several times. They closed down that aquarium, and we had a dinner in there. So when I was serving with the Defense Science Study Group with the U.S. government. And so from 1997, he was in that position until August 2024 when he retired. He received his bachelor's in chemistry from Bucknell University in 1972 and a PhD in oceanography from the University of California, San Diego Scripps Institution of Oceanography in 1979. And prior to that, he had worked for a while at Woods Hole Oceanographic Institution from 1977 till 1997. And he was part of a team of scientists and engineers that developed some in situ technology, including deep ocean Raman for in situ spectrometer, termed DORIS, enabling real-time chemical analysis in extreme marine environments. And so maybe you've wondered about how they record the structure of compounds deep in the bottom of an ocean, say around a hydrothermal vent, and they set up a spectroscopic tool, and Ed was key in the development of that. His research has advanced the understanding of ocean acidification, gas hydrates, carbon cycling. He has over 100 peer-reviewed publications and numerous collaborative projects. And so we're really excited to have you, Ed. Let me open it up to you to say a few words.
Well, thank you, Jim, for that very nice introduction. I was actually at Maillard in Moss Landing. It's not the aquarium.
Oh, okay.
People do that, they get the two confused all the time. But they do have a very nice dinner at the aquarium because the aquarium is a sister institution with Maillard. And we've had lots of receptions there over the years. And it is a very nice place for dinner. At Meillard, we have 2 research vessels. And we just got a new ship and named David Packard after our founder and financial provider. And the other ship is the Rachel Carson. When I was there, we started out with the Point Lobos, which is a much smaller vessel. We did day trips out of Moss Landing and did our experiments in the bay using a remotely operated vehicle. But the deepest it gets in the bay is about 1,200 meters. And to go offshore to the mouth of the Monterey Bay, a canyon, we would use the Western Flyer. And it had a deep diving submersible that would go to 4,000 meters. And that vessel was capable of doing, a week to a 10-day cruise. So we would go out and we'd sit on station and, launch the vehicle at 6 o'clock in the morning and hopefully had it back on deck by 5 p.m. in time for dinner. And we would spend the day playing with the submersible in the deep ocean.
So are you're in that submersible or you're controlling it from above?
No, it's a remotely operated vehicle tethered to the ship. So it has a 400 volt power supply. So power's not a problem. And that's why we were able to put a Raman spectrometer on the vehicle. Right. I would spend the day, much like today, with headphones on, sitting in front of a computer monitor, watching what's going on, monitoring various instruments. The difference being all the lights were out in the room, so we could see the monitors without reflections. So I would go to sea, spend my day sitting in the dark, watching TV.
This is interesting because a lot of young people ask me, what is my day like? But, I'm a scientist. I sit in my office a lot and I go strolling through the lab on occasion and I meet a lot with students. But there are a lot of scientists like you that are out in the field. And so if you like the outdoors, you like the ocean, you like the woods, you can still be a scientist and participate in those environments. And that's exactly where you were, right, Ed? And this is that was part of the motivation for you to become more of a chemist that worked in ocean environments.
It actually happened by accident. I was looking at different graduate schools, planning to study organic chemistry, and happened to be flipping through the University of Miami catalog. And there was a chapter on chemical oceanography, and there was a picture of a ship. And I thought, wait a minute, I could do chemistry at sea? I got to check this out. And, that's how it all started.
Wow. Let me get right to this. You worked for the famous Stanley Miller of the Miller-Urey reaction. And that had already been published, and he was already an internationally known big figure in the field. And one could even look at that chemistry, look at that reaction, and say that was probably the genesis of a lot of interest in origin of life chemistry. I personally think that was an amazing reaction, an amazing setup where he took some very basic chemicals and applied a high voltage and cycled this thing around and ended up with a lot of the amino acids. I think people have gone back and maybe they've recorded, 12 or 15 of the 20 canonical amino acids. Tell us a little bit about that experiment. I know you joined his group after that, but tell us about that experiment.
Well, actually, Jeff Bada was my thesis advisor.
Okay.
And he had gotten his PhD with Stanley Miller. And so Jeff had this idea because the Murchison meteorite has fallen a few years earlier. And a colleague of Jeff's, Keith Van Volden, was on the group at NASA that analyzed the Murchison meteorite looking for amino acids. And so there was a small group of people, and of course Stanley was involved because he knew so much about making amino acids abiotically that that's what they suspected would be happening with the meteorite. And so he was part of that all group and making, having discussions beforehand, what to look for, how to look for it. And if the amino acids were produced abiotically, what that group would look like, which ones they would find in most abundance, the fact that they would be all racemic, and that the fact that they're racemic is actually a test to make sure you don't have terrestrial contamination.
Mm-hmm. Let me just explain that. So just make sure everybody's with us. So amino acids, most of them, 19 of the 20 canonical amino acids, have a stereogenic, or what was formerly called a chiral center. And so you have a left-handed version and a right-handed version. And on Earth, from biology, from living cells, what you get is the L version. You don't get the R version. But when you just make it without biology being around, you get the D and the L version almost always in a one-to-one mixture. So if you're only getting the L version, then something might be wrong there. You might just be picking that up from some contamination here on Earth. Have I got that right, Ed?
Yes, yeah. There's actually been a lot of carbonaceous chondrites analyzed, and they find that the ones that have been sitting around in museums for decades or longer, they're covered in fingerprints. And what you find is a tremendous abundance of the L-amino acids. Right. The Murchison was unique. It fell in Australia in 1969, just before the moon landing. And so when it fell and people found it, the idea that people were going to walk on the moon, they'd bring samples back to be analyzed for who knows what. The people that found it said, we got to send this to NASA. And they took very good care when they collected it. They wrapped it in aluminum foil. They didn't touch it with their hands. And eventually it got shipped off to NASA.
And your PhD was working on the chemical analysis of that meteorite. Is that correct?
Yes, because in the Miller-Urey experiment, the amino acids are made by a pathway called the Strecker cyanohydrogen synthesis. And what happens there is aldehydes react with aminonitriles.
Right.
It's been a while.
Yeah. It reacts with ammonia to make the imine, and then cyanide hits that carbon, that imine carbon.
Yeah.
Yeah.
And so if ammonia is present, you get only amino acids. If there's no ammonia present, you only get hydroxy acids, where there's an OH group where the amine group would be in the amino acid on the alpha carbon. And if you're lucky and the ammonia concentration is somewhere in the middle, you get a mixture of the hydroxy and amino acids. And so I was taking a class in organic geochemistry from Jeff Bada the spring of my first year.
What year was that?
That was...
Was that 72?
73.
73, okay.
And yeah, it's an easy year for me to remember because that was the year I got married.
Okay.
So I had the class, I got married, and then I came back to Scripps and went to work for Jeff. And as Jeff presented it, NASA had analyzed, the folks at NASA had analyzed the meteorite, and they had found the amino acids. And the pattern was very similar to what they found when you look at the Miller-Urey experiment, where glycine and alanine are the most abundant. And as the chains get longer, the concentrations go down. And the amino acids in the meteorite were all racemic. And so, okay, there's very little contamination.
If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together, and I will share with you about why I embrace the resurrection of Jesus.
So what Jeff had proposed that we do is we analyze the meteorite and look for the hydroxy acids. And he presented this idea in class as a possible thesis project. And I was at the end of my first year. I had been working in Dave Keeling's lab, looking at CO2 in the atmosphere. And as an organic chemist, I was looking for compounds that had more than one carbon. You know, CO2 chemistry is not the same as organic chemistry. And so when Jeff made this, presented this idea in class, I went home, thought about it overnight, and the next day I went to see him, and I said, hey, if nobody else has spoken up, I'd like to give it a try. And he didn't say okay right away. He asked me how I might go about doing it. And I remembered saying, well, we would extract the meteorite the same way the folks at NASA did, and then we do cation chromatography like they did to take the amino acids out of the mixture. And then we would do anion chromatography to collect the hydroxy acids in a separate fraction. And I said, and we'll make the methylized esters using diazomethane and analyze by gas chromatography. And he sat there for a couple seconds just staring at me like, oh my goodness, this guy's actually thought about how he would do this. And we started chatting some more, but he was very slow to say, okay, we'll do it. Because I think he was also thinking about, where am I going to get the money to buy this guy a gas chromatograph? So he can do the analysis. Because Jeff's lab, they were well equipped with an amino acid analyzer, but that was all they had.
Right. Let me just point something out here. So students need to hear this, that if you go to talk to a professor and that you want to work with that professor, if you do some reading beforehand and thinking about the chemistry beforehand, and you come in and intelligently talk about these research topics, it's very different than just walking in and saying, hey, could I have a job? They're really looking for people that initiate something with some knowledge behind it, because there's a lot of students that come to us and say they want to work with us, but people who have thought about it beforehand, it makes a big difference. So that's probably the case in many professions, so keep that in mind.
Oh yeah, when somebody comes in and they've checked to see the kind of research that a scientist is doing, and if they know a little bit about it, they don't have to have it all correct, but just the fact that they took the time to read about it makes a very good impression.
Yes. So now, tell me, you spent, things have changed a lot. I started graduate school in 1981, so it was a few years after you started. 9 years or so after you started, but there was still no internet in those days. It was common to spend several days in a library just trying to find some things. We couldn't just Google it and boom, it's right before our eyes. You spent a lot of time in the library, and they used to say you spend a day in the library, it can save you months in the lab. And so we used to go to the library a lot and just go through a lot of different texts, chemical abstracts and Beilstein, but we had more time, I think, to speak between professors and students because there was no email. And can you imagine what life would be without email and all the time you spend doing email and things? So I think that Ed, what I'm trying to do is give people a picture of what life was before there was electronic mail that consumed our days. And so you probably had a lot of time to speak with Professors Bada and even Stanley Miller.
Yeah, it was a much slower process back then. And there were times when I would ask Jeff or Stanley, a question, and sometimes they answer it right away, and sometimes they say to me, well, so and so did some work on this. Why don't you go to the library and look it up? And I eventually figured out they were training me to go to the library and search for things, and back then, if you found an article that was really central to your work, the next thing you did is you went to a Xerox machine and made a copy.
People don't know what a Xerox machine is. They call it a copy machine these days. But yeah, it was named after the company that had invented xerography, which is the photocopying process.
Yeah, and they were the only game in town back then.
Yes.
And so you would make a copy yourself, and that's how you collected your background material.
Yes, and they told you so and so did this work. So much was associated with the corresponding author of the work, because there were generally only a handful of people in the world working in your area. So you could say, okay, so-and-so did this work and such did this work. And you kind of knew where they were and what they were doing, and you could keep track of all of this in your mind. Now the field of chemistry and it has exploded so much, it's just hard to know who's doing what, and people popping up all over. So it's a very different way, even than the way we used to categorize things. Wouldn't you agree?
Yeah, it's totally different. Now, if I have a question, like you say, I can take a web browser, go on the internet, type in the question, or even just type in a name, and I'll get a list of all their papers. Yes. And in many cases, you can download the ones right there as a PDF, and in seconds, you've got it.
Yeah. Now actually, I just asked ChatGPT, and it's even easier than doing Google because it sifts it out for me. But now what I want to do is look at this experiment a little bit, this Stanley Miller-Urey experiment, because you will know it very well. People think that you run this reaction and you get these amino acids, and bingo, wow, this is all great. Not only do you get racemic mixtures, so you don't get the purity of the compounds that you need to be able to make a biologically relevant type of enzyme. You get this mixture, but it wasn't just those alpha amino acids, and it wasn't just the canonical alpha amino acids. There's a lot of amino acids in there that were not relevant to life, and there were other compounds in there, and it was a selective experiment, because if you had say sugars in there as well, people don't understand. You can't deal with mixtures because molecules react with each other. If you had sugars happen to be around there, aldehydes, you would get reaction with those aldehydes, and you'd get this Maillard reaction where you would get this chemistry. You'd get an imine formation, and then elimination of an alcohol. You get these double bonds. You start getting these double bonds, and then you start getting colors after a while, and it darkens, and so also these would just decompose and go away. Tell us a little bit about this.
I call it the Maillard reaction. I don't know how to pronounce it.
I call it. I said Maillard, but Maillard. Yeah, you're probably right.
Yeah. Well, I don't know how to pronounce it because it's a French name, and so I'm probably pronouncing it just as poorly. It's a reaction that Stanley Miller actually told me about. Because, if you look up the Miller-Urey experiment, they'll always show you the equipment before it started, which is this gleaming glass apparatus that they have lit to have all these fancy reflections. And it's quite impressive. When the reaction is over, the inside of the glass is covered with this red, oily compound. And around the electrodes is a black carbon deposit. And that's where most of the carbon winds up. The amino acids they find are very dilute in the solution. They're a minor product of the reaction. And so Miller actually did the experiment over time and took samples as he went. And he noticed in the beginning, the amino acids would build up in concentration, and then they would plateau for a while. And then they would go back down at the end as the synthesis reaction slowed down. And so obviously something else was going on. And it is, as you say, the amino acids are reacting with sugars that would be produced. Now, they're not the sugars that we eat. They're much simpler. They only have 3 or 4 carbon atoms, but they are reducing sugars. They have an aldehyde at one end, and that reacts with the amine group in the amino acids and starts another chain of reactions that destroys the amino acids. You build up and you make things that are called melanoids, which just means they're darkly colored. And they have very complex structures because it's just a random combination of everything that's available. Abiogeochemists, they think of amino acids forming proteins. And you got this nice clean pathway. And what they don't recognize is the amino acids are very reactive. And they're very promiscuous in their reactivity. They'll react with anything that has an aldehyde or an unsaturation. And that means that most of the amino acids don't form proteins or even peptides because they're swept up in all these other reactions.
Yeah, that's exactly it. So it is so much fun for me, Ed, to be able to speak to an organic chemist that appreciates these sort of things. Most people think you get these amino acids and that's all that you had there. And then if you needed it to be chiral, they would selectively crystallize out. And then those crystals would find themselves all purified and ready to go. These are such sludges that they're totally unusable. And we can't even use them in our laboratory, let alone on an early Earth. And here, this being done in a pool somewhere on the side of the ocean. I mean, this is people don't appreciate that when you have messes like this, you can't do chemistry. You can't do chemistry on these wild mixtures because they start reacting with each other and the things decompose. Is that right, Ed?
Oh yeah, exactly. For my thesis research, I spent most of my time trying to separate the hydroxy acids from everything else that was there. And, I outlined the procedure to Jeff when I talked to him that first day, and I actually had to add an additional step because there were so many compounds in the hydroxy acid fraction that the peaks overlapped with the ones I wanted to look at. And so I had to break it down on size with just liquid chromatography to separate into 2 fractions so the peaks wouldn't overlap. And I could get pure peaks, and we used the mass spectrometer to look and identify them. And know that they were pure compounds.
So what happens here? It's really quite interesting. You need constrained chemistry, as John Sutherland says. You can't use what comes in on meteorites. So when people write to me, oh well, meteorites provide this. Meteorites provide. Well, first of all, they're not providing the stereo-controlled compounds that you need, but also there's millions of different compounds on those meteorites so that it becomes useless. People don't know it's there. If it's there, you can use it. No, you can't. You can't use it because of the competing reactions. So what happens is people like Ed, as graduate students, have to sit there and do chromatography. These are human-made methods to separate these one from another. And even then, it's so hard to pull these things apart, and ultimately you do lots of separations, and then you go to a mass spec, which does other separations, because what a mass spec does is it will ionize these compounds, and once they're ionized, you can deflect them with a magnetic field. And so you you change this field angle and they deflect differently, and then one comes through of a certain molecular weight, and then the next and the next and the next. And so the mass spec can pick it out. It's an amazing human tool that wasn't available on an early Earth, and it's not a preparatory tool. You're getting just a few molecules coming through at once, at each one striking that detector. And because of that, people get this false view that, oh, we saw it by the mass spec, it is there, it could be used. It was there, but it couldn't be used. It couldn't be used. And if you say, well, an early Earth figured it out, an early Earth didn't figure it out. You would have needed amazing technology to figure this thing out. And then even with human beings doing it, they just throw up their hands. These are big messes, but we have amazing tools to analyze it. Ed, am I getting this right?
Yes, you are. There was a word that they used frequently when I was a graduate student. They talked about plausible pathways. And they would look at, you can make this amino acid, it could react with another amino acid and build it up. And this was all plausible. And I was thinking about it last night, and for the first time I looked up, what was the definition of plausible? And there were several definitions. The first one is, it's something that's maybe possible, but we don't know for sure. And the second one was a little bit different, but very interesting, because it said that this is a plausible explanation is something you tell somebody to convince them that something is true, frequently to deceive them. And I thought, holy smokes. That's the whole game they've been playing, is that they find these reactions that are 1 in a million, and chances are we're overwhelmed by all the others. But there's a tiny chance that it might have happened this way. And they play this game because they're trying to convince you that it might have been possible.
Right.
Even though the odds are no.
Ed, the organic chemists get this. People who've done synthesis, they understand this. The rest of the world doesn't understand this. Biologists just don't get it because whenever they do synthesis, they buy a kit. And that kit is made by chemists. So they mix two things together. It's all loaded up for them. And they're taking their reagents from living systems. So when they have a nucleotide, it's all cleaned and ready to go. Either that's been made in a lab or it's been taken out from some other living system. And so this chemistry is so hard. And so what this does, and even before I go there, let me just continue on this Miller-Urey experiment, because you get other amines in there, you get other carboxylic acids in there. All of these are competing. And then they decompose. And people say, well, the reaction just went a little bit and stopped. But then on the other side, they'll say, well, origin of life, it had billions of years to start, billions of years. I said, if something happened and these things were generated, they don't last billions of years. Under these conditions, they last minutes and they start degrading. As soon as they start seeing any other aldehyde, they start forming these imines. If there's a hydroxyl group there, it's going to eliminate, and you'll start getting these colored compounds. That's what the redness was. And if you start putting in more double bonds, it's going to start getting more brown and eventually black. That's what these things are. It's rapid decomposition. I don't think people can really appreciate unless they've gone into a laboratory and tried to do synthesis. When you try to do synthesis and you see how hard it is with all your ingenuity, with all your modern equipment in a laboratory, and people tell you this thing happens under a rock or someplace or on the side of an ocean. It and here I am talking to a guy who's worked for the great Stanley, Miller, and Bada, and the people, this legacy. So it's not just Jim Tour making this stuff up. This is the guy who lived it. Now, tell me also, before we leave the ocean environment, tell me about these hydrothermal vents, because about 10 years ago, I was talking with a Nobel Prize winner, and he said, Jim, on your origin of life stuff that you're saying, you may be right, but you've never really considered hydrothermal vents. Hydrothermal vents have everything that's needed. And I'm thinking, what on earth are you thinking about? What do you think? Hydrothermal vents are so hot. Did you know that racemization is going to occur before you ever get polymerization? You're never going to get the polymers. So you just get the amino acids. They don't spontaneously form polypeptides. They don't, because the free energy on all of these couplings is positive. The free energy is always positive on this. They would rather be 2 molecules than 1. So entropy is terribly against you, which makes the free energy against you. So thermodynamically, you got to go uphill on all of this. And then to get these couplings to go, people say, well, you got the heat from a hydrothermal vent. I say, if you're up at several hundred degrees, you're getting racemization. So even if you had these chirally pure molecules, they're going to get all racemized. So these things are all going to mix up. You are an expert in hydrothermal vents. Ed, have I got this wrong?
No, you've got it right. Let me preface this by saying when I was a graduate student working in Jeff's lab, he had several other students that were looking at amino acid racemization. And the way they would measure the rates is they would take a water solution of the amino acid and seal it up so it couldn't boil, and they would put it in a heating block at 100 degrees or 150 degrees. They didn't go much higher than that because they were afraid it would explode. And they would make up, 10 or 12 of these tubes, and they put them all in the heating block, and then once a week or once every 2 weeks, they would take a sample out. And so they could measure the rate of racemization. Now, the reaction rate doubles for every 10 degrees C increase in temperature, approximately, roughly. That's sort of our chemist rule of thumb. If we're too lazy to use the Arrhenius equation. And the hydrothermal vents, we did some work with the ROV. We had a postdoc that wanted to study what was coming out. And so the first thing we did when we had the vehicle on the seafloor is we wanted to measure the temperature of the fluid as it's coming out of the vent. And the first time we tried it, the pilots could extend the thermal probe so far with the arm of the vehicle, but they couldn't quite get it in to the vent because they didn't want to get any closer with the vehicle. Things in hot water, especially electrical cords, don't last too long. So we abandoned that try, and on the next dive, they took a broom handle and used it to extend. They put it on the temperature sensor on one end of the broom handle, and the other end was held in the manipulator arm. And now they could get 6 feet further, and they actually got it into the vent and measured the temperature. And it's 300 degrees coming out, but inside it was 350, 400 degrees. Wow. And when they pulled it out, and it had only been in there a minute or two, the end of the broom handle was black. Well, that's odd. And then after the dive and we were taking things apart, when we looked at the broom handle, it was charred as if that broom handle had been in a fire. Wow. And, the broom handle, it's cellulose.
It's cellulose. It's made out of carbon.
So what had happened is they had dehydrated that broom handle with the high temperature and turned it into carbon, even though we were at 2,500 meters depth, 250 atmospheres of water pressure, pushing the reaction to stay hydrated. And yet they dehydrated the cellulose in that broom handle. What goes on in hydrothermal vents is not a good place for organic compounds. You'll see things come out like methane or ethane, maybe propane, but carbohydrates, no, they don't last. Amino acids, they're gone.
How about nucleotides?
Never have a chance. RNA falls apart at room temperature, right. In minutes. Yeah. So this is, there's no chance that life began at a hydrothermal vent.
Right. Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything. My faith in Jesus Christ means more to me than anything. If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandScience.org/donate. All U.S. donations are tax deductible. Thank you so much. It's such fanciful thinking, and there's all this nonsense that comes out, and sometimes Ed, I think I'm a lone voice crying in the wilderness, and people have accused me of lying. So I say this can't work. I'm accused of lying. I'm accused of many more severe things, and I don't get many organic chemists coming around me and saying, "Hey, Jim, you're right," and because you undergo attack if you say these sorts of things. If you speak the truth in science that's against the groupthink of science, you get attacked. So, Ed, be prepared to be attacked, but I guess it doesn't matter as much for you because you're retired anyway. They can't cut your grant funding.
No. I'm living off my wife's grant funding. She's got her own business, okay. So we have that coming in. But I do understand what you're talking about. There is a very strong community that is protecting the status quo. And the thing I found out early on is, if you go to any college or university, the biology faculty always outnumbers the chemistry faculty. So there's more of them than there are of us. And it's just the organic chemists that have the expertise to see the folly in some of these abiogenesis schemes. The inorganic chemists, they're not going to touch it. Mm-hmm. Physical chemists might. But the analytical chemists, they won't touch it. They
don't have the ability to really know. It's unless you've worked with molecules and you've seen how rapidly they decompose and what kind of things you have to do to get them to perform what you want them to perform, and the molecules that are life relevant. So we've talked about amino acids. Amino acids is what were made in these, and the problem is they racemize, they decompose, they react very quickly with carbonyl compounds to make imines. Those imines then start getting attacked either by a nucleophile like cyanide, or they eliminate if there's a nearby hydroxyl that can eliminate, and they start decomposing. But let's look at some of the other class of compounds. So what do you think would happen, we'll take one of the simplest ones, the lipids that people think are simple. Lipids are composed of a glycerol that is a 1,2,3-trihydroxypropane, and they'll usually have a couple of tails hanging off generally on the 1 and the 2 carbon. They'll have some fatty acid tails. These are esters. From what I know, esters can hydrolyze pretty easily, and so if they ever got near a hydrothermal vent, they're toast very quickly. And then also, if you're going to try to make those chiral, you have to distinguish between the two ends because the glycerol, the 1,2,3-trihydroxypropane, doesn't have a stereogenic center. It's only when you functionalize one end, when one end forms the ester, now you have this. They're enantiotopic, actually. The two ends are enantiotopic. So you have to selectively get one and not the other because all of these are chiral, and people have looked at these sorts of things. If you don't have them chiral, if you don't have a stereogenic center there because the middle one is going to be stereogenic, its antipode, its chirality will depend on which one of the two neighbors got functionalized, then you don't get a sufficient lipid bilayer if you don't have these chiral units there. So you have to have this chiral. So these two ends are enantiotopic. I mean, tell us about how in your mind would these ever form in a friendly way where you can make masses of these in order to make lipid bilayers?
How they form is it's a difficult question. They're very easy to take apart. In another project I did, we were looking at wax esters and triglycerides, and it's the same bond. It's an ester bond, and the wax esters, triglycerides, they're generally too long to do gas chromatography on because they weigh too much.
Yes.
So we would take them apart, and that was real easy. 15, 20 minutes with a little acid or a little base, and you take it apart. I had a friend at Maillard that used to make his own diesel fuel by taking, grease, triglycerides, and cooking it up with a little sodium hydroxide.
Right.
And he can make his own diesel fuel that way. So it's real easy to take them apart. To put them together, that is the tricky part.
That's the tricky part. And
you got to make the ester. If the fatty acids have any other functional group, you might have a reaction in the wrong place.
They compete, right?
And if the fatty acids are unsaturated, which I forget the word. We used to call them polyunsaturated fatty acids, but
yes, they could be cis or trans. Generally, we want the cis. We don't want the trans there. Yeah.
But in the health food industry, they call them something else. The omega-3s, fatty acids. Those kind of things. They're primarily made as triglycerides or diglycerides or phosphoglycerides.
And they're often allylic alcohols.
Yes.
Yeah.
And so there are all kinds of ways that the synthesis can go wrong. Now, when you have an enzyme system and things, an enzyme grabs this part of a molecule.
Now, just so that the audience knows, enzymes come when there's life. You have to first have life to have enzyme systems working. Yeah.
So your body and all living organisms can make these quite easily.
No problem.
But if you have a graduate student trying to make it in the lab, you got problems.
Right. And just reaching back now to the amino acids, half of the amino acids have active side chains. So in other words, some amino acids will have a carboxylic acid group coming off, and the other ones will have an amine group coming off. Those are indistinguishable chemically from the alpha amino acid and from the carboxylic acid where you want the chemistry to take place. So these end up participating, and those, and if they participate, it's game over. Yes, nature may use those in couplings, but that's always post-translational modification. That's modification later. You can't have that as the stringing backbone of the polypeptide. And everybody who says that they make polypeptides by hot-cold cycles, where they take a very hot surface, 150 degrees, and they'll put it on there and dry it out, and they'll make, say, an oligomer of, say, a 10-mer. They never use the amino acids that'll have a carboxylic acid hanging off, like an aspartic acid or something like that, or an amine hanging off. They always load the system because you can't do it. It can't be done. Do you agree?
Yeah. A lot of these so-called next steps after the Miller-Urey experiment, they stack the deck. They pick amino acids that will react only one way to prevent the side chains, the byproducts, the competing reactions. They make sure there's never any sugars around, because that'll go the Maillard reaction. And they tune the other conditions, the pH, the temperature, to what is conducive to what they want.
Yes. They'll go high pH, low pH, and they'll vary it according. It's all loaded, something you wouldn't have on Earth. Yeah. Okay. So we've dealt with the amino acids. We've dealt with the fatty acids making the lipids. Let's look at sugars, which are carbohydrates making the polysaccharides. Tell me about sugars. I'd seen your interview with Bill Dembski, and you talked about the foremost reaction. Then I sent you this recent paper by Krishnamurthy. And so tell me about the making of sugars.
Well, the foremost reaction was used as a plausible pathway where you can make sugars. Now, we have to be careful because sugars come in all sizes and all varieties. If you have an aldehyde and on the next carbon is a hydroxy group, you got a sugar. But it's only 2 carbons long. The ones you need, ribose, that's 5 carbons. And so you've got to add 3 more carbons with hydroxy groups to that first sugar that had 2. Each step, something can go wrong. And, the first time you add to the 2 carbon, you have 2 possibilities for going wrong. Mm-hmm. But then with 3 carbons, you got an aldehyde and 2 hydroxy groups, you have 3 chances to go wrong. And when you get to 4, now you have 4 chances. And so the number of reactions that don't go to where you want to go doubles with each carbon that you add to the sugar. So it's hard to make sugars non-enzymatically.
Yeah. And in the foremost reaction, you get billions of compounds. People don't appreciate that when you have a billion different compounds that all have the same types of groups, they all have hydroxyl groups, they all have an aldehyde group and things like that, that you can't ever do chemistry on that mixture because other things would compete. They think if you can see it by mass spec, it is therefore there and you can use it. You can't use it. You have to have more constrained chemistry. Wouldn't you agree?
Well, you can do lots of chemistry. Very little of it is what you want. I can tell you sort of a story of my early in my career when I was a synthetic organic chemist. I did very well in class. The professor picked me up to do a summer internship with him, and I was very good at following the recipes for making the reagents that we wanted to use. I could do those kind of reactions. But then when it came time to try something new, to do some new chemistry, starting out with pure reagents. So we've eliminated all the side reactions, but only what's going to happen with our reagent and the compound that we want to either oxidize or reduce. And invariably, every one of my reactions were turned into this gooey mess. And the reason I got so good at analytical chemistry was I was trying to find out, well, what went wrong? What did I make instead of what we thought we were going to make? And I would have these very complex mixtures of organic compounds. And I do IR and NMR and, trying to figure out. And that was the beginning of my end as an organic chemist, because I turned out to be a better analytical chemist dealing with the messes I was making than, and all my colleagues that were working on the bench next to me, they were doing different reactions with different compounds. And they get done, they'd have these beautiful crystals. They take an NMR or an IR and they get beautiful spectra, and I had messes. And it was like, it was frustrating. But in the process, I was learning how to use all kinds of analytical instruments.
Right.
Which, when it came time in graduate school, I was set.
Yeah, it served you well. And that's what you developed to look into the hydrothermal vents, to look in the deep sea chemistry. Yeah. I
remember from this paper, there was an NMR spectrum that was purporting to show ribose.
Yes.
And where the peak was supposed to be was a lump. Well, who knows how many compounds were there, but it wasn't the nice sharp ribose peak. And I'm like, well, what's going on here? Because those are the strong peaks they should be looking for. And I'm thinking to myself, they didn't show up.
The spectrum that was put out by Benner for ribose, I brought that to a carbohydrate chemist. And I said, would you look at this spectrum? And I said, would you call this ribose? He looked at it, he says, this is garbage. This is garbage. You can't do anything off of this. No, that's not just noise. This is trash, and you can't do chemistry. Now, we'll go to the next step, the polymerization of these. So if you take, for example, 6 glucoses, people have done the calculations because 6 glucoses, so you'll have these hydroxyl groups on glucose. You have a number of hydroxyl groups here, and then you have this aldehyde. This can form a 6-membered ring or a 5-membered ring, or it can be an open form, and you have all these different alcohols. So if you take 6 glucoses, these can be hooked up. People have done the calculations, 1.2 trillion ways, 1.2 trillion ways that you could arrange 6 glucoses. This is crazy. If you have 6 A bases, I mean, how many ways can you hook this together? I mean, A. Now you can have, if it's on RNA, you can have the 2', 5' linkage, you can have some branching. But sugars are so hard to deal with. So if you can't hook these up in the right way, you have both the alpha and the beta at the anomeric center, but then you have all the other regioisomers. Making the polymers and all of those polymerization steps, every coupling is a free energy that's positive. So making the polymers thereof, some people have said, are even harder than making the small molecules, and that may well be the case. It doesn't look like we have a solution to the carbohydrate problem, to the sugar problem, does it?
No. You'll make lots of products, but none of them are what you want. Yeah. And if I remember my sugar chemistry correctly, and now I had the class over a half century ago. So the difference between cellulose and amylose, I think it is an alpha-beta linkage.
Exactly. You remember it properly. It's the alpha-beta orientation at the anomeric center that does this.
Our bodies make amylose to store the glucose for use later. We can't digest cellulose.
Right.
We can't eat wood.
People don't eat wood. People don't eat grass. Cows eat grass because they have an enzyme. They have a microbe in their gut that can actually break that linkage for them. Yeah.
And that just tells us how specific the polymerization has to be.
Yes. So people realize this is just something that's either this way or this way. It's just up or down. You get that wrong, you can't eat it.
Yeah. Well, you can eat it. You just not going to do
it goes
good.
right through you. You don't get any advantage from it. It doesn't become part of your body.
They call it fiber.
Yeah, it's fiber. So we can't make the amino acids or the polypeptides. We can't make the fats. We can't make the sugars. There's one class left. That's the nucleotides, the DNA and RNA. Now, a nucleotide has to be made up of a sugar. So if we can't solve the sugar problem, we can't solve the DNA RNA problem, right?
No.
All right. So we can't make any of these things. And people say, oh, well, did you see that paper? They're taking these trimers of nucleotides and they're doing this. I said, well, wait a minute. If you're really telling me this has relevancy to early Earth, where did they get their nucleotide? And in every case, they got their nucleotide. It came from a natural source. It already came from life, from living biological system. So of the 4 classes of compounds that build us, Ed, you and I have come to agreement that we can't make the small molecules that make them up, nor can we do the polymerization. Because when you do the polymerization, say of RNA, you can have the 3' 5' linkage, which is what you want, or you can have the 2' 5' linkage, which is what you don't want, and you can have branching off that extra hydroxyl, plus the instability, as you were mentioning, that extra hydroxyl just swings around and hits the phosphate of the one lower to it, and the thing falls apart. So we can't make the small molecules of the 4 classes of compounds. We can't make the polymers that generate these. We're kind of lost on origin of life, aren't we?
Yes. The Miller experiment was a tremendous advance because it was the first time somebody stopped talking and actually went in the lab and tried to do something. And it created a lot of excitement. But it's been 75 years and we haven't gone very far.
I say this all the time, man. I am so glad you said it before I did. You said it before I did, and this is a guy who worked with Stanley Miller. So we can't get these compounds. And what my argument has also been is we may be able to approach the target a little bit more, but what happens is the target moves much further away from us because we learn the complexity of what is in a cell. So, for example, there's something called the interactome. This wasn't known until about, 25 years ago, this interactome, just the non-covalent interactions, the way things are lined up. The interactome just in a simple yeast cell for just protein-protein interaction, that has 10 to the power of 79 billion. That's a 1 with 79 billion zeros after it. That many possible combinations, and only a few of them are going to work. So you learn these sorts of things. The other thing is chiral-induced spin selectivity, which was discovered about 20 years ago. This chiral-induced spin selectivity, which tells us that reaction chemistry has to have the right configuration on the helical structure, on the enantiomeric structure, because you get electron transfers down these chains, and without that, you get too much heat release. And so you couldn't have had life unless you had that first control. So it's not like enantiomer structures could have evolved to have pure enantiomers later. You couldn't have done this with the racemic compounds. It had to be at the start of life. So all of these problems, that's why my argument is every year when we learn more about the complexity of a cell, the target to make the simplest cell has now become harder. So in essence, we're moving further away.
Yeah. The amount of discoveries in biochemistry that have happened since the Miller-Urey experiment, changes the game. It's so much more complex than the target they had back in mind back then. Mm-hmm. If I remember correctly, I think the structure of DNA was announced
right about
the same year.
Yeah, right about that time.
As the Stanley Miller, 1953. The target that Stanley had when he started his experiment, they didn't even know what DNA was. And that's how juvenile the field of biochemistry was. Mm-hmm. It's grown up a lot.
It's grown up a lot.
And with each new discovery, the target for abiogenesis gets further away.
Gets further away. And when they discovered that structure of DNA, they didn't really understand the code. That came about 3 or 4 years later, where they figured that you had these trimeric units that are defining for a specific amino acid, and that's how the information is now transferred. I mean, it was they knew so little compared to what we know now about a cell, and we continue to learn so much, and you've got to solve every one of these problems. And people say, well, cells were much simpler back then. And so biophysicists have back calculated what is the simplest cell you could possibly have. And you still have 15 different massive structural components that we have no idea how to make. None of them. And so we're so far from being able to solve this thing. It's a big problem, this whole origin of life problem. And for me as a scientist, I could never sit here and say it will never be solved because I don't know what people are going to know in 1,000 years. I don't know. But as far as we know, we're not even close to solving this in our own laboratory. Putting aside origin of life, this is how life formed on an early Earth. Just saying, can we even take life? We can take a cell, as you know. We can take a cell, deconstruct it, and have all the different polymers there. Have the DNA there, so we already got our code. Have all of that code and everything. We can have all the carbohydrates in their polymerized form as the polysaccharides. Once it's apart, can anybody take those parts and put them back together into a living cell, Ed?
You can't put Humpty Dumpty back together again.
You can't do it. And it's not even that we can't. We wouldn't even know how to go about doing it. You put all these things together in a liposome, they don't all start functioning. It's like taking all the parts of your engine and throwing it inside throwing it on the driver's seat and saying, hey, if I shake up this car enough, it's going to start going. It doesn't go. And so we have no idea. So let alone how it happened on an early Earth, we can't even do it in our modern laboratories. This is how clueless we are on origin of life. Before I wrap this thing up, Ed, I want to ask you a question because I've heard about your background. Do you believe in the physical resurrection of Jesus Christ?
I do.
Good. So do I. And this is what I want young people to see. This whole thing of the resurrection of Jesus Christ is based upon historical evidence that man has risen from the dead. People can have very strong and productive scientific careers and still be believers in Jesus Christ and walking with him as Lord and Savior. And this is what I want people to be able to see. Ed, I want you to wrap this whole thing up for us. Is there anything that you want to say on origin of life or on faith or any matter you want to talk about?
One of each. Origin of life. I once asked Stanley Miller how long he thought the prebiotic soup was on Earth. You've got an atmosphere of reactive gases. It's reducing. There's lightning. How long did that situation last? And because that sets the limit for how long you have to make the first living organism, or puts one limit on it. And what he said totally surprised me and caught me off guard. He said 10 to 50,000 years. You don't have 100,000. You don't, maybe 100,000, but you don't have 1 million. And 1 billion years is totally out of the question. You've got a very limited time for where you've got the prebiotic soup and you've got all this work to do. And every time they make a new biochemical discovery, you got more work to do. Mm-hmm. So that's what I would say.
That's good. That's good, yeah.
So
time... We don't have the time that people think we have.
You don't have the time.
Yeah.
And time doesn't help you. Because you and I both know if you're doing a synthesis and it doesn't work, you don't come back the next day, set up the same experiment and say, I'll give it a month, and come back and expect to get a better result.
Right.
The scientific revolution began with the astronomical revolution. Copernicus, Galileo, Kepler,
right.
Newton. They were all Christians. They all believed in the physical resurrection of Jesus Christ.
Yes.
They did pretty well in terms of science. I think we're in good company.
We are in good company. And the reason, people have gone back and they said, why did this come from this Judeo-Christian background? And the idea is that we expected God to work on regular and consistent means. It's not like Poseidon is upset today and therefore the winds are going to come. We don't have a capricious God. We have a God that is reliable. We have a God that is sound. And so we can expect that what we do today, if we duplicate it, it's going to be the same tomorrow and the next day and the next day. You didn't have this in other faiths. You didn't have this in other considerations. And here you have this. And so there was this expectation that we have a God that is regular in the sense that there's consistency. We have physical laws that are consistent. And that was the origin of all of this. Well, thank you so much, my friend. For me, this has been really exciting because I feel like I have touched history. And so for me,
Are you saying I'm an old guy?
well, you know, that you interacted with Stanley Miller of the Miller-Urey experiment, which was 75 years ago, was this experiment, and that you had worked with him and talked with him and interacted with him, and now I get to interact with you. I mean, this to me is like sort of like these Erdish numbers of what's the distance from these Genesis points. But for me, it's a very big deal. Thank you so much for coming on this channel. I know you don't normally do these sorts of things, and so for me, it was great having you. And I suspect there's going to be other people that are going to come knocking on your door and want to get an interview with you because they're going to want to touch history as well.
Well, thank you very much. This has been a lot more fun than, well, this is my first web interview. So I had no idea what to expect, how it was going to go. And it's going very well. I've enjoyed it. Thank you very much for inviting me. And hopefully we've shed some light.
Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.